Welcome to AP2. For our first lecture, we're going to pick up with Chapter 15, which covers the autonomic nervous system. Now, before we get into too much detail with the autonomic nervous system, we are going to do a comparison, comparing the somatic nervous system, which is where we left off last semester with the neuromuscular junction and action potentials propagating down neurons, and then leading into this division called the autonomic nervous system. So let's start by comparing the somatic and autonomic nervous systems. The somatic nervous system is going to include both sensory and motor neurons, similar to what we saw in our pathway in AP1. Sensory neurons are related to those peripheral sensations like touch, pain, temperature recognition, and proprioception, which is a sense of balance or self-position. We also get our special senses like sight, taste, smell, and equilibrium or balance that's detected in the ear. The motor neurons are going to innervate skeletal muscle and is going to affect or give us a response to the effector, the muscle, in response to those senses. Now, the autonomic nervous system is going to be a little bit different. It receives input from sensory receptors located viscerally in our organs, blood vessels, muscles, specifically smooth muscles, and the nervous system. So we're going to use a few pictures to help compare the somatic and autonomic nervous system. So now this should look familiar. Now, we remember that when we talked about reflexes and the somatic nervous system, we talked about a sensory stimuli coming in through the dorsal root, coming through here, synapsing with an interneuron here, and then sending information out the ventral root to the effector. Now, what we're going to focus on is we're going to kind of bypass this portion right here. So we're going to skip this and focus right here when we look at the autonomic and somatic nervous system, and we're going to see exactly what that motor neuron looks like, what it does, what it touches, and how it works. So if we look at our somatic nervous system here, we see a couple things of interest. Number one, it is a multipolar neuron that's leaving the central nervous system and moving out that ventral root, going to the effector. We have a single axon. That axon is myelinated, so we see that there is myelin here. We also see that it's going to extend to skeletal muscle, right? That's why it's somatic, goes to muscle. The neurotransmitter being secreted at those synaptic end bulbs is acetylcholine. Okay, so a couple things. Again, just to reiterate, one axon, one neuron. The neurotransmitter is acetylcholine, and the effector is skeletal muscle. Okay, so again, this is a key component of the somatic nervous system. That brings us to the anatomy of the autonomic motor pathways. Now, before we look at a picture to compare, let's look at some details of the autonomic nervous system. Now, most autonomic motor pathways, again, remember we're skipping the sensory pathway and the integration pathway. We're going right to the motor pathway. These autonomic nervous system pathways consist of two motor neurons, so that's key. We're going to put a big star and put two motor neurons. Now, the motor neurons are named. They're named as a preganglionic neuron. Now, the preganglionic neuron, notice the word preganglia. So we're going to have a knot of neurons at some point that forms that ganglia. So the neuron before the ganglia is preganglionic. It's going to have its cell body in the CNS, that central nervous system. And the axon is going to extend to that autonomic ganglia. The second neuron comes after the ganglia and is called a postganglionic neuron. Now, the postganglionic neuron has its, look at this big word here, unmyelinated axon extending from the ganglia to the effector. Okay, so a couple key points to recognize. 2, remember those numbers. 2, that's going to be a trend through the autonomic nervous system. 2 motor neurons, one preganglionic, one postganglionic. One's myelinated, one's unmyelinated. Now, let's take a look at some of our options for our autonomic nervous system. We're going to use the diagrams to kind of look at differences between some of these pictures here. So let's take a start and look at number 1. Let's see what we see. So again, remember we're looking at the motor neuron. So our preganglionic neuron, we'll keep it here. Preganglionic neuron, green. Our preganglionic neuron, look at it, it's myelinated. Okay, so number 1, it's myelinated. Number 2, so we'll write myelinated. Number 2, it's relatively short. So the preganglionic neuron, preganglionic, is myelinated. It's short. When we look at the ganglia, the neurotransmitter it's secreting is acetylcholine. Okay? Now, let's take a look at the post. And again, let's just reiterate that's number 1 here. Number 1. We'll look at number 2 and number 3 as we continue on. So the post-ganglionic neuron is going to be 1, unmyelinated. Number 2, it's relatively long, okay? And 3, the neurotransmitter it's secreting is going to be norepinephrine. So our neurotransmitters are going to be a little bit different. So again, look at this post-ganglionic neuron. It is long, it is unmyelinated, and the neurotransmitter is norepinephrine. Now look at the effectors. Glands, cardiac muscles in the heart, smooth muscles in the bladder, digestive system, things like that. Okay? Now, let's see exactly the difference. We're going to move on to number 3 here. So let's move on to number 3. Before we get started, we're going to erase the information on number 1 and then rewrite it. So number 3, let's see what we see. Let's look at the preganglionic neuron. What do we notice? 1, it's myelinated, so it has myelin. 2, it is much longer than the preganglionic neuron in number 1, so that's a little bit different. Number 3, the neurotransmitter it's secreting is acetylcholine, so there's some similarities between 1 and 2 so far, just in the fact that the preganglionic neuron is myelinated and at the ganglia they're both secreting acetylcholine. One major difference between 1 and 3 is the length of the preganglionic neuron. Number 1 is short, number 2 is long, which brings us to the postganglionic neuron. Now that postganglionic neuron we see is unmyelinated, so there is no myelin there. It is relatively short in comparison to number 1. And number 3, the neurotransmitter secreting, in stark contrast to number 1, is acetylcholine, whereas number 1 was norepinephrine. So, before we talk about number 2, let's look at these differences and why this is happening. So, why are there differences between number 1 and number 2? Well, they're different divisions of the autonomic nervous system. So, let's look at number 1 here. Number 1 is sympathetic, fight or flight. Number 2 is parasympathetic. So, some differences. Why? These two divisions are foils. They're not really antagonists. They accentuate each other. When one is activated, the other is suppressed, right? They're not really canceling each other out. Again, not antagonists, they're foils. If you think about foils in novels like Ron and Hermione, if you want a good spell, you count on Hermione, whereas Ron would be scared and cowering in the corner, right? So again, why these differences? So when we talk about what they do, look at the effects. They're the exact same effectors in both systems. But again, remember, one is fight and flight, one is rest and digest. Again, these are foils. We're going to get into a little more detail as we continue on with our lecture. Now we can look at number 2. We kind of saved the middle of the road, the oddball out here for last. Now, let's look at what's going on here. Look at the preganglionic neuron. It's very similar to one. So let's go preganglionic neuron, right? It's very similar to one. But wait a minute. We're using this word preganglionic. We know that the autonomic nervous system should have 2 neurons. This one doesn't. It only has 1. So there is no preganglionic neuron. We have a special circumstance here. This is a special way that we get sympathetic outflow, where we do not utilize 2 motor neurons. We're only using 1. So let's look at this 1 motor neuron. It is myelinated, so it has myelin. It is going to be relatively long because it has to go from the spinal cord to the adrenal medulla. Okay, so we're not going to see a short and long. It is part of the sympathetic division. So that's a little odd because we're typically seeing in the sympathetic division, like above, that we have a short preganglionic neuron and a really long postganglionic neuron. Here, we get 1 neuron. It is myelinated. It goes from the central nervous system directly to the adrenal medulla, where it's secreting acetylcholine. Okay, and then the result are going to be the cells of the adrenal medulla, the chromaffin cells. So here's those chromaffin cells are going to secrete norepinephrine and epinephrine, aka adrenaline. So this is odd. So here we see a couple things going on. Number 1, we see norepinephrine twice. Once as a neurotransmitter, twice as a hormone. Are they different? Nope. They're exactly the same. They are structurally the same. They're just named differently because they're working in different capacities. Again, one is working as a neurotransmitter. The second is flowing through the body, through the blood as a hormone, but structurally the same component. Now, why do we have this? This is our direct fight or flight response. When we get scared, when we're in frightening situations, we need a fast response to that stimulus. So we get this special circumstance where we're going to bypass the chain ganglia, so we eliminate the ganglia, bypass the chain, and go directly to the adrenal medulla to get a really fast, all-encompassing, entire body fight or flight response. So now that we covered some basic anatomy of the autonomic nervous system and its 2 divisions, let's put it to type. So the autonomic nervous system is divided into, again, 2 divisions. The sympathetic division is our fight or flight response because it stimulates an increase in alertness and an increase in metabolism to be ready for frightening, harrowing, and emergency circumstances. The parasympathetic, the second part of our division, the parasympathetic nervous system is referred to as the rest and digest division. It is also responsible for reproduction and repair as it stimulates slowing down most body activity. So we're allowing for heart rate to slow, breathing to slow. We're going to activate, suppress all our other fight or flight systems and activate our repair reproductive system for reproduction, our repair system for fixing body, bones, tissues, cells, and we're going to stimulate digestion. So we look to table 15.1. This is a nice little comparison of the somatic and autonomic nervous system, looking at sensory input, control of motor input, the motor neuron pathway, neurotransmitters they secrete, the effectors, and responses. So again, everything we've talked about in the past few slides in a nice, neat little package. So let's continue with the anatomy of this autonomic motor pathway. Remember, each division of the autonomic nervous system has 2 motor neurons. The preganglionic neuron, which has a cell body in the brain or spinal cord, and our postganglionic neuron that has its cell body and dendrites located in the autonomic ganglia, so in that ganglia, where it synapses with its preganglionic axon. Now let's take a look at the sympathetic division. The cell bodies of preganglionic neurons are located in the lateral horns of the gray matter in our 12 thoracic and first 2 or 3 lumbar segments. So we have another name for this division. It is called thoracolumbar. So this is another way that we can explain the sympathetic division. Thoro for thoracic, co meaning with, the lumbar vertebra. So again, another name for sympathetic autonomic nervous system, thoracolumbar. Now our next slide is going to give us a picture to see what we're looking at here. So let's put it all together. Again, thoracolumbar, we're dealing with the thoracic vertebra and the first 3 lumbar vertebra. We see our short preganglionic neuron. Here is our chain ganglia, right? And our long postganglionic neuron. Now let's stop for a second and just kind of put this to an analogy to understand why there's these anatomical differences between the between the sympathetic and parasympathetic nervous system. So think of it like this. Why short preganglionic and long postganglionic in the sympathetic? Why long preganglionic and short postganglionic in the parasympathetic? Why are they opposite? Well, we're going to use an analogy, and I hope you guys can relate with this. The analogy is going to be Batman. So if you want to call Batman, how do you do it? Right? So if you want to get Batman's attention and say, hey, come talk to me, we have problems, we use the bat signal. Now, what is unique about the bat signal? The bat signal can be seen by, you guessed it, everyone. So that's kind of like the sympathetic division. It is essentially the bat signal. So when that sympathetic signal goes out, it casts a net far and wide. So why is that important? That's important because when we have a stressor, a flight or fight response, we are activating everything. We are sending a signal far and wide. We're talking to everybody here. We're talking to the eyes, the lacrimal glands, the heart, the lungs, the liver, the stomach, the small intestine, the large intestine, the kidneys, the bladder, the genitalia. We're talking to the skin. We're talking to everybody all in one fell swoop. So we get an all-encompassing response. We're activating everybody, putting everybody in the pot, and making one big soup. So that is unique to the sympathetic nervous system. Now the parasympathetic nervous system is different, and we'll see that in our next few slides. The parasympathetic has these really short postganglionic neuron, and the reason being is because we are not casting a signal far and wide. It's actually the opposite. This would be like Commissioner Gordon calling Batman on the bat phone and having an intimate conversation with him saying, hey, how's Alfred doing? Have you seen Catwoman? So the parasympathetic has this intimate communication with each organ, again, the same organs we see here, intimately. So parasympathetic can activate just the urogenital organs, just the digestive organs, just the heart and the lungs, just the skin, right? It can activate specific parts because it has its postganglionic neuron inside the near or inside the actual effector organ. So 2 different types of command signals for different types of modalities. Next is the parasympathetic division. Now, the cell bodies of the preganglionic neuron are located in the nuclei of 4 cranial nerves, 3, 7, 9, and 10, in the brain stem, and in the lateral gray matter of sacral segments 2 through 4 of the spinal cord. Now, we're going to give another name to this division as well, and it is called craniosacral. So kind of like thoracolumbar, we have craniosacral here because we are identifying that we are in these first 4 cranial nerves, 3, 7, 9, and 10, and in the second, third, fourth segments of the sacral portion of the spinal cord. So again, craniosacral is our other terminology. Let's take a look at a picture of this, and here we go. Again, this picture does some justice to what we were just talking about, right? So look at this preganglionic neuron. So on some of these, we follow that preganglionic neuron all the way down, and look, the postganglionic neuron is short. Look how short they are. They are close to or even embedded in the organs, in the effector organs, again here and here. What this does is it allows the parasympathetic division to say, hey, we just ate a meal. Let's activate the small intestine, the large intestine, and start with digestion. Okay. At the same time, we don't want to be stimulating the reproductive organs to be making, say, gametes or things like that. So the parasympathetic allows us to pick and choose who we want to activate during these times of rest and digest. So a unique difference to that all-encompassing casting that wide light, like that bat signal, to tell everybody to come play or get into the game. Here we can pick and choose who we want to activate. So little important distinction. Now, if we look at it from a clinical perspective, we say, well, stress is essentially a fight or flight response. Is this a good or bad thing? And the answer is, it really is a bad thing, because remember, when we are activating and stimulating the sympathetic, the parasympathetic is suppressed. So essentially we could draw a hand and we can say that the parasympathetic system is depressed as the sympathetic system is accentuated or activated, and vice versa. When the sympathetic system is depressed, the parasympathetic system will be activated or accentuated. Again, this is a balance, a teeter-totter all day long, as these guys work dynamically all day long to flip between sympathetic and parasympathetic responses. As we continue with our anatomy of the autonomic motor pathways, I want you to remember our rule that we have through this lecture, our rule of 2. And as a review, there are 2 autonomic ganglia, sympathetic and parasympathetic. We have 2 major subtypes of sympathetic ganglia. The first being the sympathetic trunk ganglia. This is also known as the chain ganglia. It lies in a vertical row on either side of the vertebral column, which gives us our prevertebral ganglia, which lie in front of or anterior to the vertebral column and close to our large abdominal arteries. The next step in looking at our autonomic nervous system, specifically the motor pathway, is to look at our sympathetic outflow. So exactly what happens to information leaving the spinal cord. So after axons of the sympathetic preganglionic neurons enter sympathetic trunk ganglia, they may connect with postganglionic neurons in one of 4 ways. So we have 4 ways that we get autonomic or sympathetic outflow. Number 1, an axon may synapse with a postganglionic neuron in the first ganglia it reaches. Number 2, an axon may move up or down, so may ascend or descend that chain to a higher or lower ganglia before synapsing with a postganglionic neuron. Number 3, an axon may continue without synapsing to the chain through the sympathetic trunk ganglia to end at a prevertebral ganglia and synapse with postganglionic neurons. Number 4, an axon may also pass that chain without synapsing through the sympathetic trunk ganglia and a prevertebral ganglia and extend directly to the adrenal medulla. That was that weird circumstance that we had if you go back to number 2 on our 3rd or 4th slide, where we talked about the only having one motor neuron leaving the central nervous system and going directly to the adrenal medulla right here. Again, super important to give us a really fast fight or flight response. So again, 4 ways to get sympathetic outflow. In our next slide, we'll look at a picture to kind of explain this a little better. So let's put what we just talked about to drawing. Number 1, we can leave the spinal cord, go directly to the first ganglia in the chain that we see, and move directly out to our effector organ. Number 2, we can leave and move either up the chain or down the chain to a subsequent ganglia and then move out to the associated effector organ. Number 3, we can leave the spinal cord, bypass the chain, go right to a prevertebral ganglia, and go to our effector organ. Number 4, we can bypass the chain and go directly to the adrenal medulla, allowing us to get a single neuron to activate the adrenaline flight or fight response to the entire body hormonally. The next anatomical structure of an autonomic motor pathway we have to talk about are autonomic plexuses. They are formed from sympathetic postganglionic axons, parasympathetic preganglionic axons, and some visceral sensory organs. These plexuses are unique because they provide this complex innervation pattern to our target organs. Now remember, since most organs are innervated by both divisions of the autonomic nervous system, we're going to find these plexuses will include places that are aligned along or following major arteries to which they are distributed. These are going to include things like the celiac plexus. You may have heard of this. It's commonly called the solar plexus. We follow the superior and inferior mesenteric arteries to form the superior mesenteric plexus and the inferior mesenteric plexus, the renal plexus, and the hypogastric plexus. So we can look at a picture, follow some of our major arteries, and find our plexuses. Follow the pulmonary plexus by the pulmonary trunk, the cardiac plexus by where our heart would be. We have an esophageal plexus by the esophagus. Let's follow some major arteries that we just talked about. Here's our celiac trunk, our celiac plexus, superior mesenteric and inferior mesenteric plexus by the superior and inferior mesenteric arteries. Renal plexus by the renal artery, hypogastric plexus in that hypogastric region. If you go back to AP1, where we have the regions and quadrants, this region is the hypogastric region, so hence we name it the hypogastric plexus. Now, a couple things to identify too. We have our chain ganglia, so that lies on the sides of these vertebrae. Okay. So again, just pointing out some structures that we have already identified. So I know this is a lot of information, so throughout this lecture we're going to pause and do some reviews just to catch up and make sure we're all on the same page. So let's relook at our sympathetic preganglionic neurons and their cell bodies. I can't stress to you the importance of recognizing and realizing where they start and where they end. Remember, the cell bodies of our sympathetic preganglionic neurons are part of the lateral gray horns of all 12 thoracic segments and the first 2 or 3 lumbar segments of the spinal cord. The paired sympathetic trunk ganglia are anterior and lateral to the vertebral column. Usually there are 2 cervical, 11 or 12 thoracic, 4 or 5 lumbar, 4 or 5 sacral sympathetic trunk ganglia, and 1 coccygeal ganglia. Now to review our parasympathetic preganglionic neurons, cell bodies are located in the nuclei, in the brainstem, and in the lateral gray matter of the 2nd through 4th sacral segments of the spinal cord, and cranial outflow for parasympathetic extends from the brainstem and cranial nerves 3, 7, 9, and 10. Now again, we didn't really talk about these cranial nerves, but if you go back to AP1, cranial nerve 10, the vagus nerve, is one of the most important cranial nerves in the body as it touches almost all of our visceral organs. So we will revisit this again. And then sacral outflow extends from the 2nd through 4th sacral spinal nerves. Remember our terminology, craniosacral. Now let's take a closer look at that parasympathetic motor neuron. Look how long that preganglionic neuron is. Look at the postganglionic neuron, intimately tied inside that organ. That's that key difference between parasympathetic and sympathetic innervation, right? We see that we can activate just the bladder when we need to and not worry about this all-encompassing activation. We don't want everything parasympathetic, rest, digest, repair, and reproduction activating at once. We need this intimate connection to talk to one versus the other. The next step in this system is to look at the neurotransmitters and their receptors. Again, follow your rule of 2 as we travel through our next few slides. So our autonomic nervous system, neurotransmitters, and receptors, based on the neurotransmitter they produce and release, we're going to look at autonomic neurons are considered to be either cholinergic or adrenergic. Now let's look at the terminology. Cholinergic, choline hints to acetylcholine, adra hints to adrenaline, right? So let's look at cholinergic neurons release the neurotransmitter acetylcholine. We could break these down into subsets of two, meaning nicotinic and muscarinic. The second subset is our adrenergic neurons, which release norepinephrine or adrenaline. So again, subset of two neurotransmitters, two classes, cholinergic neurons that release acetylcholine, and they can be either two types, nicotinic or muscarinic, which we'll get into in our next slide. Our class of adrenergic neurons, they are going to release norepinephrine or noradrenaline. So let's take a look at our different types of sympathetic and parasympathetic neurotransmitters and their receptors. So we start with the receptors. The nicotinic receptor is a channel protein that upon binding of acetylcholine, it opens, which is going to basically allow diffusion of cations. The muscarinic receptor, which is a little bit different on the other hand, is a membrane protein. Upon stimulation by the neurotransmitter, it causes the opening of ion channels indirectly. So this works through a second messenger system. So muscarinic receptors are going to be a little bit slower in response than nicotinic. Now, nicotinic receptors are key players in neuronal communication, converting neurotransmitters binding into membrane electrical depolarization. Now, muscarinic are going to be a little bit different. They're going to be activated by muscarine, whereas nicotinic are going to be activated by nicotine. Now, let's take a closer look at these nicotinic receptors in the sympathetic division. One that's going to utilize an adrenergic receptor on the effector cell, and one that's going to utilize a muscarinic receptor on the effector cell. So right here at the preganglionic neuron, we see a nicotinic receptor being stimulated by acetylcholine, which is going to activate that adrenergic that postganglionic neuron to secrete norepinephrine, which will bind to the adrenergic receptor on the effector cell. And we know it's adrenergic because it is responsive to adrenaline or norepinephrine. Now, on the flip side, we look at the muscarinic receptor cell. So let's see, we start with a nicotinic receptor on the preganglionic neuron. There's our acetylcholine again. We activate the postganglionic neuron, which is going to secrete now a little bit different acetylcholine, right? Instead of norepinephrine, because these are cholinergic receptors, which is going to bind to that muscarinic receptor to activate that second messenger system. So we see this innervation to most sweat glands in the body. Now for parasympathetic division, a little bit different. We start with our nicotinic receptor. There it is, secreting acetylcholine in that really short postganglionic neuron, which will then secrete acetylcholine again. So we have a cholinergic receptor binding to a muscarinic receptor on the effector cell. Again, the second messenger system. So let's look at a table that kind of puts it all together, 15.2. So for our cholinergic receptors, their major locations are integral proteins and postsynaptic plasma membranes activated by acetylcholine. So for nicotinic, its location, plasma membrane of postganglionic sympathetic and parasympathetic neurons. Its action is excitation, so sending impulses in the postganglionic neuron. Chromaffin cells of the adrenal medulla. Remember that special way we bypass the chain and go directly to the adrenal medulla. Effects is release of epinephrine and norepinephrine. We can go to the sarcolemma of skeletal muscle fibers at that motor end plate. Think back to that neuromuscular junction. These nicotinic receptors are going to excite the muscle to give you your contraction. Muscarinic, remember, slower. These are second messenger systems. The effector is innervated by parasympathetic postganglionic neurons. Its response is going to be in subreceptors excitation and in others inhibition. Sweat glands innervated by cholinergic sympathetic postganglionic neurons. The response will be increase in sweating. Skeletal muscle blood vessels innervated by cholinergic sympathetic postganglionic neurons. The effects will be inhibition to relaxation via vasodilation of your blood vessels. That brings us to our adrenergic receptors. Location, integral proteins in postsynaptic plasma membranes activated by the neurotransmitter norepinephrine and the hormones norepinephrine and epinephrine. Now, we are going to get into different types of receptors, alpha 1, alpha 2, beta 1, beta 2, and beta 3, but we're not going to go crazy with these. I don't want you to memorize the specific different types. Just know that these play a major role when we talk about indications for drugs like for high blood pressure. We talk about beta blockers that are blocking these receptors from being bound, which will decrease force and rate of contraction of the heart. So there's that beta blocker, so it blocks beta 1 receptors. We can talk about beta agonists, so we can talk about these beta 2 receptors. If you have asthma and you take albuterol, these, instead of blocking B1 like we saw in that blood pressure medication, these activate beta 2 receptors, which cause a relaxation and dilation of your airways. So this is good if you're getting inflammation and closure of that bronchial tree during an asthma attack. This kind of opens everything up so you can breathe easier. So beta blocker here, blocker for, again, the clinical connection drugs, and this is an beta agonist, so stimulates that receptor. So our next few slides kind of summarize everything that we've talked about. So neurotransmitter summary, some general statements. All preganglionic fibers for the sympathetic and parasympathetic nervous system release acetylcholine at their synaptic terminals. Important point. At the ganglionic synapse, the postsynaptic acetylcholine receptor is a nicotinic receptor. The effect is going to be excitatory, resulting in propagation of that action potential or signal. Our postganglionic parasympathetic nervous system or cholinergic response receptor fibers also release acetylcholine. At the effector tissue, it could be either excitatory or inhibitory, depending on the particular receptor. Ultimately, most but not all postganglionic sympathetic nervous system or adrenergic fibers release norepinephrine. There are some circumstances where they will release nitric oxide, but most will release norepinephrine. So the nice thing about this slide where we think twos, I kind of been hinting this to you all lecture long, so we could put everything we've talked about, all 27 of these slides, into one nice neat little package. Think twos, 2 neurons, preganglionic myelinated and postganglionic not. 2 neurons, 2 neurotransmitters, acetylcholine, preganglionic, and norepinephrine, postganglionic. 2 exceptions, some postganglionic neurons secrete acetylcholine and nitric oxide instead of norepinephrine. 2 divisions, sympathetic and parasympathetic, with 2 origins. The sympathetic nervous system is thoracolumbar, and the parasympathetic division is craniosacral. 2 types of adrenergic receptors, alpha and beta. 2 subtypes that we introduced clinically, but you do not need to know the specifics of these adrenergic receptors, alpha 1, alpha 2, beta 1, and beta 2. And 2 types of cholinergic receptors, muscarinic and nicotinic. So again, a beautiful, nice rule of 2 to get you through the autonomic nervous system. The last few slides deal with the physiology of the autonomic nervous system, and for this we go to a few tables. The first table we look at is 15.3 that compares and contrasts sympathetic versus parasympathetic divisions, its distribution, location of preganglionic neuron cell bodies and outflow, where the associated ganglia locations, axon length and divergence, white and gray rami communicantes, whether they have it or not, neurotransmitters, where they are and what they do, and physiological response. So again, puts everything we've talked about into a nice, neat little table. Table 15.4 goes through the effects of the sympathetic and parasympathetic divisions of the autonomic nervous system, so what each has on specific organs, like the glands, adrenal medulla, the lacrimal glands, the pancreas, posterior pituitary, pineal glands, sweat glands, adipose tissue, liver, kidneys, and juxtaglomerular cells within those kidneys. So again, we can read through the effects and chart down. Notice that the sympathetic has more effect on these organs for fight or flight than do parasympathetic, which only affect a few: secretion of tears, digestive enzymes, and again, no known effect, and then we hear some glycogen synthesis and bile secretion. So parasympathetic mostly, again, rest and digest and digestion, right? Digestion is where we see activation here. We continue with the visceral effectors like the heart muscle, the eyes, the lungs, the gallbladder, stomach, spleen, ureters, sex organs. So all those organs that these systems touch and what they do. You should have a general understanding, again, not focusing in on the specific alpha 1, alpha 2, beta 1, beta 2 receptors, but basically what they do, right? So for example, for the sympathetic cardiac muscle, we increase heart rate. And then here's that teeter-totter. For the parasympathetic, we decrease heart rate, okay? We can go to the eyes. The sympathetic is going to, the ciliary muscles of the eyes are going to relax to adjust shape of the lens for far vision. For rest and digest, we're contracting for close vision. We look at the lungs. During fight or flight, we're relaxing to get more air, more oxygen to sustain the fight or flight. And for rest and digest, parasympathetic, we're contracting to get less air so that we can divert to other places. Stomach and intestines, fight or flight, decrease motility and tone, whereas for obviously rest and digest, parasympathetic, we're increasing motility. So again, go through these. A lot of them make sense. I gave you a few examples, but you can go through and see how they balance each other out for each organ system. We continue on with visceral effectors like vascular smooth muscle, heart, skin, skeletal muscle, etc. So you can move your way through, continuing through these tables and what exactly each system does. The next and final step of the autonomic nervous system is examining the integration and control of autonomic functions. Integration and control. Functions such as heart rate and force of ventricular contraction, blood pressure, and blood vessel diameter are controlled by autonomic reflexes that occur when nerve impulses pass through an autonomic reflex arc. Now, the reflex arc is composed of 5 steps. Number 1, a receptor. 2, a sensory neuron. 3, an integration center. 4, a motor neuron. And 5, an effector. We will spend some time later on in our next lectures discussing and detailing the 5 steps of this reflex arc. So again, just a basic introduction. Hang tight onto this as we will get into a little more detail in our upcoming lectures. Our second to last slide looks at our focus on homeostasis, just like we did in AP 1. We look at what this autonomic nervous system and how it affects all the different systems of the body. So you can read through this little chart and see how we balance a stable but not static environment in our body. Lastly, we talk about disorders. We start with autonomic dysreflexia. This is a syndrome in which there is a sudden onset of excessively high blood pressure. Now, this tends to be more common in people with spinal cord injuries that involve the thoracic nerves of the spine or above T6. The second disorder we're going to look at is called Raynaud's syndrome or Raynaud's phenomena. Now, this disease causes some areas of your body, things like your fingers and toes, to feel really, really cold and numb in response to cold temperatures or stress. And we can see this when your fingers, lips, toes, and hands and feet could turn really, really white. In Raynaud's syndrome or disease, smaller arteries that supply blood to your skin tend to narrow. This limits blood circulation to those affected areas, essentially causing these little vasospasms. Now, the interesting note about Raynaud's is that women are more likely than men to have this syndrome. It appears to be more common in people who live in colder climates, and that makes sense, right? Because with the cold we see the onset of this disease. People always ask, well, how do I treat Raynaud's syndrome? Well, it depends. It depends on the severity and whether you have other health conditions. For most people, this disease isn't disabling, but it can affect your overall quality of life.